and Associate Editorial Board Member, Current Medical Imaging. Dr. Ravindra Hegde completed BSc in 1991, MCA in 95, MPhil in 2003, and PhD in 2003 from Gulbarga University. His areas of research are digital image processing, medical image analysis, document image analysis, biometrics, machine vision, robotics. He has guided 10 PhD students and 4 students are currently pursuing their research under his guidance. He has published 179 research papers. He is member of Indian Unit for Pattern Recognition and Artificial Intelligence, member of International Association of Engineers, member of International Association of Computer Science and Information Technology. He has authored a book titled Medical Image Analysis, published by Lambert Academic Publishing, Germany, Multilingual Document Image Analysis, published by Lambert Academic Publishing, Germany. Recent Trends in Image Processing and Pattern Recognition, Part 1, 2 and 3, published by Communication in Computer and Information Science, Springer Nature, Springer Private Limited, Germany. He has carried out many sponsored research projects, consultancy work, he has organized many national and international conferences and seminars. He is member of Board of Studies and Board of Examination for many universities and institutes. So I once again welcome you, sir, on behalf of our institute.
Thank you, madam. Shall I continue, madam?
Sir, you can start your presentations.
Okay. First of all, I would like to thank the principal, my best friend Mallikarjun Hadi sir, for giving me an opportunity to deliver a talk. In fact, I am delivering 2 talks, one on medical imaging and another one on CT scan image segmentation and all. So I also would like to thank the college for giving me an opportunity. Today in this session, I am going to talk. I hope okay, I will start my presentation. Hope my
Sir, it's not visible, sir.
Is it visible now, madam?
Yes, sir.
Okay. So now I am going to deliver a talk on the medical imaging research avenues. So in this, I have divided this talk in 2 parts. In the first part, we will see what are the possible medical imaging techniques that are available where information technology, software, IT machines as well as software development is involved, where the researchers can find an avenue to do some research. And later on, we will look at certain challenges where there are many challenges that we have where we can do some research. So here I am going to present you some challenges where these challenges are actual challenges that are available. There is one medical imaging grand challenge site. From there I have collected many challenges. So in these challenges, they will provide you the images, they will provide you what kind of problems that are required to be solved, they will provide you the domain knowledge, all that. So these information will be provided, and you are, if you are interested, you can take up this challenge and you can do or you can solve certain problems. So let us look at what is medical imaging technologies. So basically, medical imaging technology is used to produce images of organs and tissues in the body for the use in diagnosis and treatment. Suppose, for example, we are using so many technologies such as MRI, CT scan, X-ray. Many technologies we are using. Certain technologies are totally based on the IT. For example, MRI, CT scan and all, they generate 3-dimensional view of our body. So these are totally dependent on the computer developed programs. That is the technology what is being developed by the IT people who are working in the biomedical engineering field. So these basically help in the diagnosis and treatment. Basically, we are not going to diagnose. We are only engineer technicians who help the medical fraternity, and medical fraternity will use our developed technology for further treatment. So basically, we can call it as a type of diagnostic testing, that is conducting a kind of test before conduct of any surgery or before conduct of any further treatment to the patient. So here we can find different types of medical imaging depending on the kind of the body part. For example, if doctor want to look at inside the body, like the soft tissues, they use a different modality. If they want to look at the bones, they use a different modality. If they want to look at brain, they use a different technique. If they want to look at the stomach, they use a different kind of technique. So we have so many kinds of techniques depending on the kind of the part of body which we want to basically look, or which doctor feel like looking. So diagnostic imaging tests provide more information as compared to the other tests. So because here basically we are going looking at inside the body, so we will go inside in depth inside the body and we are looking at. So definitely this gives us some more information as compared to just testing over the body, where doctor just look at on the body and do certain tests. So actually they provide the visual pictures, they provide the structures of the body, and they also provide functioning of organs, how actually organs are functioning, whether they are functioning normally or whether they are functioning abnormally, whether they are not functioning, whether they are slow functioning, fast functioning. All these informations are being provided through the diagnostic imaging. So these technologies are some of them are quite expensive. Like if you go for MRI, CT scan and all, they are very expensive. There are certain technologies such as X-rays and all, they are low expensive. But definitely when we look at compare with the other kind of testings that doesn't involve these kind of technology, definitely these are more expensive. But the effectiveness of these is very good. Okay, even though it is limited because of its properties, but the technology what we use, that is going to help a lot to the physician to take up further treatment. So that's why nowadays you can find that many of the doctors they recommend for doing any such kind of testing before going for before recommending any further treatment. So now we will look at what are the different modalities that are there before going on to the kind of research problems or kind of the challenges that exist. So one such technique is called as endoscopy. Most of you must have undergone the endoscopic technique. So in the endoscopy, basically, endoscopy involves it will have a very long scope. It is a kind of tube, and in that tube there are many instruments also fitted. At the tip of here you can find at the this is a scope which goes through endoscope, which goes through mouth. Okay, it is passed through mouth, and basically the endoscope is used to check the any diseases that are there in the entire this tube. Okay, what we call it as esophagus as well as the stomach. Okay, this entire tube. Suppose for example a patient is suffering from certain cancer in the esophagus, then through endoscope we will be able to visualize it. And if there are certain ulcers in the stomach and all, even we can pass this scope. It will be very long. We can pass this scope through this stomach. And at the tip of the endoscope, there will be light source. You can see here, there will be light source. So inside the stomach, there will not be light. So if you want to capture any image, you need light. So this light source will focus, okay, the light over the tissues which we want to observe. And it will have other instruments such as some small tube will be there. Suppose, for example, there are certain food stuff remaining in the stomach. We want to see behind that food, whatever deposit has happened. Behind that, is there any kind of a disease, something like that? So for that, there is a tube which pumps the water and flushes and cleans that region. Suppose we want to take out the water from the stomach. Okay, there may be excessive water. Okay, so we want some time to take out that water. Then there is a tube which sucks the water. Okay, and there are certain small instruments which can take the biopsy. Biopsy means the sample of that particular part. Suppose there is a tumor, the doctor want to check whether it is a cancerous tumor or normal tumor and all. So doctor can pick that part, so some instrument to take the biopsy also. So these kind of techniques are involved in the endoscopy. and it will have a very important part, that is camera. So this entire thing will be lively visualized over the screen. Okay, doctor can see whatever the technique he is performing, the movement of the scope, entire thing, the scenery inside the stomach can be visualized using the camera, which will be connected to a display unit. And even he can record, he can capture the snaps and all. These all are possible in this endoscopy. Then we have thermography. The thermography basically is used to check the body temperature of the body temperature. Suppose, for example, let us assume in certain part of our body blood flow is slow, okay, or blood flow is blocked. So when blood flow is blocked, then what happens? That part will get paralyzed, and basically the temperature in that part, due to the non-flow of the blood, the temperature will drop down. So we can check where the blood flow is not proper, where there is a blockage and all. Particularly, it will not give you perfect blockage idea, but where in which part of the body blood flow is not there, that can be visualized with the thermography technique. Then we have very important, that is X-ray. Generally, everyone must be aware of X-rays because everyone must have gone X-ray and undergone X-ray many a times. So X-ray is basically it is a high energy electromagnetic radiation which are passed through the human body, and wherever there are bone parts will block the X-rays, and the soft tissue part will allow the X-rays to penetrate through body. So there will be a film behind our body, and in that film, whatever the rays will be developed in the dark room, and then we will get a picture like what you have seen, what you can see on the screen. So here clearly we can visualize whatever the breakages are there in the bone and all. Particularly this is used to check the breaks in the bone. Sometimes accident may happen and all. At that time, to check whether any where exactly the breaks are there, where exactly bone has cracked, how many pieces it has broken. To check all these, it is used. So X-ray basically it is a non-invasive. Non-invasive means we need not put inside the body anything. Okay, without touching the body also we can take the X-ray, and it is painless. It will not create any pain and all, and it will happen within a fraction of a second, very quick. Okay, so and then the exposure to X-ray, repeated exposure to X-ray is also not good, because that can cause the repeated X-ray can damage the cells inside the body, and that can lead to the development of cancer in the later stage. So that's why the doctor suggests don't go for X-ray many number of times. Okay, don't go for X-ray for oftenly. Then we have another modality which we call it as body scanning, body scanner. So body scanner is used to check entire body, particularly the skin cancer and all. This is also emitting a very high sensitive radiations. So this is also dangerous for the health. But if somebody is carrying any weapons, any kind of narcotic material inside the body, you must be knowing nowadays people use all kind of techniques. Okay, they are not they are ready to take the risk of their life also, but they do these kind of drug trafficking, okay, gold smuggling, all these. So if they are carrying something inside the body also, then to find that we use body scanners. So these are, you can find such body scanners in the airports and all, where the people will be checked for entire body. They will be scanned for entire body. Suppose the security people feel that there is a suspicious things in a particular person, then they will undergo the body scanning. Generally, everyone will not go for body scanning, but whenever there is a suspect, they will go for body scanning. Then we have the computer-aided tomography. So this revolutionized the entire visualization of our body in the form of a three-dimensional view. So CT, we can sometimes it is called as computer tomography, computed tomography, and generally its long form is computer assisted tomography, CAT, but in short we call it as a CT scan. So basically this CT scan uses an X-ray itself. Okay, X-ray itself is used, but multiple times X-ray is taken, and based on that it generates a three-dimensional view. So about CT scan, in my next session, I'm going to explain more because our work is based on the CT scan image only. I will just briefly explain here. So it helps in construction of a three-dimensional view. Okay, and this three-dimensional view, we can go inside the body. In X-ray, what happens? It is a two-dimensional view. But in the 3D view, we can be able to see every part of our organ. That is the, suppose we are looking at the bone, we can just look at the bone in a sliced manner. Or we can even look at the other parts of the organs by slicing every piece of, I mean, not actually physically slicing, but the conceptually we will slice every part and we will be able to see inside the human body. So it may be used for diagnosis of the diseases such as the cancer or any skeletal abnormalities there, or any vascular diseases are there, such as the blood flow problems and all. These can be used. So since it uses the X-rays itself, this is also very harmful, and you are not supposed to undergo this treatment oftenly. So it is very harmful. Then we have the next modality, that is fluoroscopy. So fluoroscopy basically uses a kind of X-ray material. Okay, so this is injected into inside human body. Suppose assume that in my body, in my for example, in my hand there is blockage of blood flow. With the previous technique, we can only see that blockage is there in the hand, but where exactly blockage happened, identifying that is difficult with the previous technique, that is thermography. But we can use fluorography. So fluorography, in the fluorography, what happens, the patient usually have a ingest or a contrast liquid. Okay, so that liquid will be put into inside the nerve. Okay, so this liquid actually contains barium or iodide. Okay, these two, one among these two material. And then when the liquid goes inside the body, then they will go for a technique called as angiogram. Okay, so they used to study blood vessels of heart and brain. This dye is injected. This is called as a dye. The material containing barium or iodide. This dye is injected into the blood veins, and when it flows wherever there is a blockage, it stops over there. Okay, and then this will help us in identifying where it has stopped. And you know, fluoroscopy, fluorescence, it will emit the rays, and that rays will be captured and it will be looked at or studied to understand where blockage is happening. And then we have ultrasound. So many of you are very much aware of the ultrasound. Ultrasound actually is majorly used in the medical segment, but ultrasound has many other applications also other than the medical. So what is this ultrasound? Ultrasound, a device basically uses the sound frequency. A high frequency sound waves are used here. Basically the device produces these waves, and these waves are passed through the body where you want to check. Particularly this is used to check the health of the child inside the womb. Okay, inside the pregnant woman. Okay, so many times it is being also misused to check the sex of the child. That is illegal and it is punishable, but this is very much essential to check the health of the child and to plan the proper treatment by a doctor. So what happens here, the rays goes and wherever there is a change in the tissue, okay, wherever there is a change in the tissue, there rays will get reflected. Some rays will get reflected. So likewise, the high speed rays will go further inside, and wherever there will be change in the body, like the mother's body, and then there is a liquid, and then after that child's body starts, and then child's body ends, and then mother's body starts. So like this, many rays we get. I am audible, no?
Excuse me, sir.
We will get many waves. I am audible,
Sir, you
na?
are audible, but it is echoing, sir.
Voice is echoing,
Yes,
madam?
sir. Yes, sir.
Just a minute.
Okay, sir.
Now, madam, is it okay?
Yeah, fine, sir, fine.
Okay. So basically what happens here, okay, better I will use the headphone, I think.
Yes, sir.
That will be,
It will be fine, sir.
okay. Okay, somebody can respond whether sound is echoing now?
Yes, sir. No, sir. Perfect, sir.
Okay. Okay, okay. So I was talking about the ultrasound. In the ultrasound, basically, what I was telling is wherever there is a change in the body from child to mother and in between so many layers will be there. So there the rays get reflected and reflected rays are captured by the ultrasound device, capturing device, and that will be connected to computer, and computer will generate a three-dimensional view of the image. You can see here in the picture there are two images. One actual image is there, and then using software techniques it is being reconstructed. You can see some color development. A 3D view is generated for visualizing the child. So these two images you can see, actually this is the first image, is the actual image, and then after reconstruction using software techniques, the second image you can find. This is ultrasound. Then we have MRI, so magnetic resonance imaging. So MRI is also very one of the important technique, because this also is used to develop the three-dimensional view. If we compare the MRI with the CT scan is used to look at the hard tissues, whereas MRI is used to look at the soft tissues. CT scan uses X-ray and it is harmful, whereas MRI uses magnetic signals, so it is not that harmful as compared to the CT scan. So it uses basically strong magnetic and radio waves to generate and these waves interact with the hydrogen atoms in the body, and that's how the rays what are passed through the human body will be captured at the other end, and then the three-dimensional view of the body is generated. So a computer basically uses these signals, whatever the signals that are received from the magnetic waves, okay, from the source of the magnetic waves, and then a three-dimensional view is generated. The reconstructed image reconstruction happens using the in the computer. So used to image, it is used to image the structure and function of the brain, heart, soft tissue, and inside the bone. So the main target of MRI image is the soft tissues such as the brain, okay, and then heart. Also people use MRI for scanning the heart. CT cannot be used for heart scanning, okay, or heart reconstruction, whereas MRI can be used for heart reconstruction. Then any other soft tissues of the body, or inside the bone. Inside the bone we have soft tissues, what we call it as a bone marrow. So to scan them we can use MRI technique. Then this is used to diagnose cancer, then brain disease, and also problem related to the circulatory system, blood flow system and all. Then we have the positron emission tomography, PET. Okay, in short we call it as a PET. We know short abbreviations, so the long form also I am telling you. Positron PET is quite expensive, and it uses the nuclear medicine. Okay, it is very harmful. PET is very harmful because here we use nuclear material, and you all know nuclear material is very harmful. They emit the gamma rays, beta rays, gamma rays, all those which are very harmful for any human body. So PET scans are a type of nuclear medicine in which the patient is given a radioactive isotope that emits the positron radiation. So the patient will be injected with the radioactive material. Entire body will become radioactive, okay, and the patient will himself or herself will start emitting the X-rays, the gamma rays, that is radioactive rays. So radioactive isotope is attached to a chemical absorbed by these certain tissues or organs. So here what happens, these tissues and organs will absorb these radioactive material, and then they will start emitting. So based on the emission signal, whatever the signal that get emitted, the machine devices are fit which will capture these rays, and they will be able to tell is there any problem, anything like any cancer tissues are remaining, something like that. This technique is used to analyze the small variations also. It will give very small, suppose a small cancer tissue is there still in the human body, this technique will be able to it will able to identify is there any small cancerous region inside the body. So it is used to detect cancers, heart diseases also, and even in some brain disorders such as we call it as Alzheimer's disease, which basically the patient lose the memory problem, or those kind of things we have seen in many of the movies. So this is very harmful. This is expensive also, and this is a very perfect test that are being currently used by many of the therapists. So then we have radiotherapy, what we call it is in short radiation. Generally you must have heard. So generally radiations are used to kill the cancerous tissues. Okay, but it is so dangerous that it also kills the good tissues also. When you go for a radiation, when any patient go for a radiation, particularly the patient which is who are affected by the cancer, they go for undergo the radiation. When they undergo radiation, the radiation kills the cancer tissues, but also it kills the other normal tissues. So radiotherapy is used use of X-rays. So it uses the X-rays to kill the tissues. Basically, it is used to treating the cancer. So X-ray damages the DNA of the cancer cells. Okay, either kill them or preventing them from multiplying. So it will kill the DNA in the cancer cell, or it can disable the cancer cell DNAs. But beams of the X-rays, how they do it, they send the beam of X-rays over the affected part of the human body, and then at the tumor or affected part, wherever you feel like in this position cancer was there, and after performing surgery to kill the remaining cancer cells, they use this particular technique. But they always damage the healthy tissues also surrounding the bad tissues. So this is often combined with other treatments such as surgery and chemotherapy in the medical segment. And then we have a nuclear medicine. So this is also something like the previous one. This uses the radioisotopes. So in the previous one, entire body will become nuclear. Here I think they provide the images of how tissues and organs, basically particular tissues if you want to look at, then they use nuclear medicine. So basically in this, the radioisotope produces the radiation and it attached to a chemical that is absorbed by the body. So here also they use the same radiation technique. As the radioisotope emits radiation, a special camera and computer detects its the images. It captures these emissions and it produces the image. And it is used to diagnose the cancer and to investigate the blood circulation and even to evaluate the organ disorders. Then it is also used for treating some cancers like prostate, thyroid or breast cancers also. Then we have one more technique which is very less known to us. It is known as biophotonics. So in the biophotonics, most of us are not aware about this. It is a light is beamed on the cells and tissues. Okay, a kind of light, biophotonics, in the biophotonic techniques, a kind of light is beamed on the cells and tissues, and light is scattered by the molecules of these cells, and special device is used to record these scattered patterns. So as we focus the light on the tissues, then that light gets scattered, okay, from those tissues by hitting at the molecule of the cells, and then there will be recording devices which record these images and they produce the image. So these are some of the modalities through which the images are captured in the medical segment for the treatment and analysis and treatment. Now we will look at what are the challenges. There are some challenges. We will look at the kind of challenges. These are basically the current issues that are being used by many of the people.
So
there is
once again, once again, sir.
yeah. Okay. So
Okay.
we will look at these challenges, and we will be able from this you can even select one or the other problems as you, if somebody is looking for the research. Basically, I hope many of you are either doing PhD or you people are looking for doing PhD. If you are willing to do PhD in medical imaging and all, these challenges will definitely help you to understand the current research problems. And even I am also planning to put two challenges in this. I'm planning to put one presentation I will give you next, and then we are working also on the knee, that is osteoarthritis also. I am planning to go for one more challenge, two kind of challenges I want to put in this. So basically the medical image, medical image processing is one of the very vast challenging area. Many people across the world are working on medical imaging problems. Many companies are working on that. Many companies are developing devices embedded with the software for working over the medical images. So this is a very big industry. Okay, medical imaging is a very big industry where many people are working on this. Many courses are being run. Many universities are providing degrees on this. So this is a very challenging as well as vast, and it is never ending kind of a field. So many researchers are working across the global on this area, and then we will see some of the challenges which are being currently attempted by many of the researchers. So there is one challenge which is put in the medical imaging grant challenge. So you can visit later, you can afterwards you can visit a website called as medical image grant challenge. Just put the Google search as medical image grant challenge. You will get one website there. These challenges are there. I have picked from there itself, and I am producing you. So one challenge is there on pathology visual question answering. So in this challenge, basically it is here you need to develop a deep learning model. Okay, this you need to use the deep neural network models for interpreting pathological images and question answering. I put certain question. Okay, by looking at the image, it has to give me answer. Okay, so this is what actually is expected in this. So the task is to given a pathology image, they will give one image, okay, and the question are given. Then you have to check whether these questions, that means whether the image contains a cancer cell, you have to answer. Okay, you have to look at the question, you have to check what is the question, and accordingly based on the image content you have to give the correct answer. So actually they provided also the data with this challenge. There are around 4,998 images, and they have even given the questions also. 32,799 questions are there. So along with the images. So on average for each image there are around 6 to 7 questions. For every given image there are 6 to 7 questions. So what you need to do if you want to accept this challenge, you have to take the images along with the questions, and then you have to check, or you have to provide the answer for that image. You have to develop the learning model, deep learning model, and then you should be able to correctly provide the answer for the questions. So I have even given you the site address. You can just look at this site address to look at more details of this particular challenge. In every slide I have given the site address, so you can visit later. I will provide the PPT also. You can visit later to this site and you can check the challenges. Otherwise, you can go visit the grand challenge. There, every challenge you will be able to find. Then we have another challenge, that is quantification of ultra uncertainties in biomedical images. So in this challenge, it is to benchmarking algorithms that quantify uncertainties in the biomedical image segments. So uncertainties basically means sometime what happens, there may be a good tissue, you may doctor is unable to judge whether it is a good tissue, bad tissue, or there may be bad tissue and doctor will be not in a position to tell it is a bad tissue. So these kind of uncertainties you have to work on, and you have to develop the technique. So here basically you need to use some binary segmentation techniques, and then the various pathologies and anatomical structures such as brain, kidney and prostate in MR or CT images data you have to identify. You have to find the uncertainties in such images. So here they have provided the challenge details as well as the images in the site address, what I have mentioned over there. Then one more we have, that is cranial implant design. So suppose for example a patient undergoes some kind of accident and all, and the accident due to hitting of the head, and there is a crack in the head or inside the brain, they want to operate, cut the part of the head, hard part of the skull, and then they want to do certain operation and all. At that time this is required. So cranioplasty, this technique is known as cranioplasty, is a surgery process where a skull defect is repaired using a cranial implant. So defect in the skull part itself, okay, that will be treated. You can see in the image, this skull part is cut apart and then they have done the implant over here. Okay, so what are the challenges here? The task or the challenges involve obtaining the 3D view of the data of skull with the defect, that is they will provide you the CT as well as MRI images, and you have to they are 3-dimensional images, and then you have to segment out, okay, converting the 3D image data into 3D mesh model you have to generate, okay, you have to create a mesh model for that, then creating an implant. So from this mesh model, you have to generate an implant for that, okay, where the disease part is there. For that part, you have to generate an implant, and then you have to provide that information, implant information, to the challenge who has provided the challenge. So for this, they have provided 200 high-resolution images, 3-dimensional high-resolution images that are 215, 259, 3D means the width is 250 and height is 250, and then there are many such images depending on the requirement. So there are such 200 3-dimensional images. So the image site is also provided here. Then we have lung nodule analysis. So basically lung is our one of the body part. So in that sometime the cancer get developed, so we call that as a lung nodules. So lung cancer is a leading cause of cancer related deaths across the world. Many people die because of the lung nodule cancer. So this needs a treatment. For that the CT can be used, MRI can be used. So in a CT lung cancer screen, many millions of CT scans will have to be analyzed. So when you want to look at this problem, they will provide you many CT scan images. So you have to scan those images, and then you have to identify in which image or in which CT 3D image the lung nodule is found. Therefore, there is a lot of interest in develop the computer algorithms and to optimize screening. So basically, because of this challenge, many images are given. Many people are planning to working on this problem. So the LIDC or IDs are RI data. So okay, they have collected some data and they have given some name, LIDC, ID, RI, maybe based on their lab name, something like that, or the developer's name must be there. So this data is publicly provided over here. So you can use this data, and they also have annotated data, including the annotations of the nodules by 4 radiologists. So annotation means they will take help of the radiologists, the doctors, and they get it annotated. Annotated means putting the boundary over the cancer region or the region where you want to identify. In the image you can see here, there is a nodule. Okay, this must be a CT image. There is a nodule, and you can see that part is visual, looking like a white part, okay, which may not be normal. So your task is to identify. They will provide you the annotated images, okay, and same images they will provide you without annotation. Then you have to look at the annotated image, and then you have to develop the algorithm such a way that it has to capture exactly same as the annotated image. So this is the challenge over there. So generally, whenever you go for any such challenge, generally you will get the images which are annotated. So that means the image which are annotated, they will give both annotated as well as the normal images. and same images they will give you annotated. Why? Because why we need annotated means because we cannot say where the lung nodule is there. We cannot decide. Okay, we are only the computer engineers. Okay, we can only look at the exact image where the nodule is there, and then we can work on our algorithms, and then we can compare whether our results is perfect or not. So in any medical imaging, you need annotated image. Whenever you want to work on any medical imaging, you need annotated image. That is very much needed. Then we have cranial, okay, so we have looked at this. Then we have another challenge, that is skin lesion analysis towards melanoma detection. So this is a skin related disease which we can see over the using any normal cameras. We don't need any high-end devices for capturing this skin diseases. So the goal of this problem is to develop image analysis tools to enable the automated diagnosis of melanoma from dermoscopic images, that is skin image. Melanoma, it is a skin disease, and when this skin disease occurs, you have to segment this lesion part. What are the analysis you need to do? Basically, the problem is divided into 3 parts. So the skin lesion analysis problem is divided into 3 parts. The one is lesion segmentation. You have to segment that part, okay, wherever the skin disease is there. Then detect and localization of visual then you have to localize means you have to identify only that part which is having the disease. Then you have to extract their features, okay, how it is looking like, because there may be variety of melanomas. In the melanomas there may be 10 to 15 or many number of types of diseases. So we have to identify which type of disease exactly it is, and then we have to classify. We have to extract the features from that, and then we have to classify those features into different disease categories. So this is the problem involved in that. And then at the bottom, I have given you the website address of this particular grand challenge. Then we have the endoscopic vision. In endoscopic vision, we have just looked at what is the endoscopy. Endoscopy, it's a invasive, that is, it has to be put inside the body, and they capture the images from the inside the body. So here, instrument segmentation and tracking also is sometimes required. That is, where instrument is moving, okay, whether it is the tube what we have put, what the doctors have put inside the mouth. So see here, endoscopy is one technique. There are other techniques also, laparoscopy, then bronchoscopy, then we have so many, 5 to 6 techniques are there. Endoscopy is put through mouth, okay, whereas the one more technique is there, I think laparoscopy, that is put through nose also, okay. And then you must be knowing there are other parts of body where we can put the tube, okay, wherever there is a hole. Different techniques, different modalities are used to look at what is the problem inside that particular part. So then this here basically tube tracking, where exactly the movement is happening, to track that, and segment and tracking is also one kind of the challenge over here. Then detection of cancer, early cancer, okay, early stage of cancer, okay. So inside when you look at, you can see here there is one tumor kind of region you can find. In the second image, tumor. Actually in the first image, actually this is a cancerous part, because by looking at the image itself, it looked like there is a cancer region, okay, major severe cancer is there inside this. So such tumors you can find inside that. So you need to identify whether it's a good tumor or bad tumor. Sometimes there will be good tumors also, okay, which doesn't carry any cancer. But if there is a bad tumor, then you have to identify that, and at early stage you have to identify, and then you have to help the doctors to conduct the surgery. So abnormal detection in gastroscopic images. So this entire part is, this is esophagus, then stomach, this all we call it as a gastroscopic part. So the images will be captured, video will be captured, and then it will help us in analyzing. Then workflow detection from laparoscopic images. So these are some of the challenges that are provided in this specific image modalities. So you can find these grand challenges in the website what is mentioned over there. Then we have gland segmentation. So gland are important for human bodies, okay. So gland are important histological structures that are there in the human bodies. Then the main mechanism of these glands are they secrete proteins and they secrete carbohydrates. So because of the proper functioning of these glands, our human body grows, our all structure, everything properly grows. If any particular gland is not properly functioning, then that particular gland which is reason, for example, you can find very tall persons, so excessive gland secreting happens, or very small person or something like that. Some people will have some problems, some people in the early stages their skin will look white and all. So these are all because of non-proper functioning of some of the glands inside the human body. So malignant tumors arise from the glandular epithelium, okay, also known as adenocarcinoma, are most prevalent form of cancers. So in this gland also some cancer happens. Okay, we have to identify whether the gland is affected by any kind of cancer or not. Okay, so this is the kind of challenge over here. So what are the challenges here? The gland segmentation in colorectal adenocarcinomas, and then gland segmentation in breast adenocarcinomas. These are some of the challenges that they have provided here. So they provide you the images, they provide you the information regarding these problems. There you have to develop the algorithms to identify these problems and to give the solution from the problems. Then we have another problem known as ischemic stroke lesion segmentation. See, many problems are there. Some of the problems are being produced over here in the grand challenges. So across the world, if you look, many people are working on many problems. So we can understand what kind of problems actually people are working, and then we can attempt our own, or we can take those challenges and we can attempt them also. So this ischemic stroke lesion segmentation is a subacute ischemic stroke lesion segmentation is required over here. Then acute stroke outcome, penumbra estimation, these are the kind of requirements that are there in this problem, and the details of this is provided. Ischemic, it is related to the brain. Okay, this is a disease related to brain. Here I think they must have provided you with the CT, the MRI images. These look like a MRI images, and they have given 3 images in this figure itself. You can see in the photograph. The first image is the first day of the problem, that is captured in the scanned whenever they scanned. Okay, whenever patient came with a complaint, they have scanned it, and after 3 days again they scanned. That is second image. You can find the white part is developed here. White part means it is a kind of hardened part inside the body. And then in the 28th day, they again scanned, performed this MRI, and you can see that region has become larger. Okay, the disease has grown up over there. So this you have to track the growth, you have to identify, track everything you have to do in this particular challenge. Then we have the retinal OST cyst segmentation. So in the retina, there is a disease where the cyst is get developed. Okay, cyst is a bad tissue in the retina. So here we develop that is STOCT, spectral domain optical coherence tomography. Retinal cyst segmentation images are given, and you have to develop the algorithm to segment those spectral domain optical coherence tomography images. So they have also provided the annotated images. That is the images. You can see one annotated image is provided in this figure where the cyst part is annotated. Okay, rest of the part is normal, and only the annotated part is actually the cyst part. Okay, so by looking at these annotated parts, you have to work on the other images, and you should be able to use this intelligence to segment the other parts. So this is actually annotated parts are images are given by the experts at the Vienna Reading Center. So this is a research center, and Optima at Medical University of Vienna. Okay, so 2 institutions have joined together to develop a challenge for this. They have provided you images, they have provided you the kind of challenges that are there, so you need to attempt over them. You can visit the website address given over here to attempt your problem. Then we have the diabetic retinopathy detection. So inside the eye, retina is there. So as the person become aged, okay, so the diabetes get developed in the retina, okay, and that's why you can see the eye surgery happens after certain ages. So if we don't go for eye surgery, then visual capacity will come down, okay, it will lead to the blindness, okay. So we call this as a diabetic retinopathy. So it is one of the leading causes of the blindness in many of the people, particularly in the aged population across the world, and many people get affected because of this, and most of them, maybe around more than 90% people get affected due to the diabetic retinopathy. So around the world, if we estimate, the current population which is affected by this is 93 million people. So currently detecting diabetic retinopathy is a time consuming process, and it is being done manually. So this needs the automation, or it needs the development of computer algorithms. I think many people are working on it, and many people must have also achieved the good rate of accuracy in this particular domain of problem. So here what you need to do is a trained clinician to examine and evaluate digital image fundus photographs of the retina are already provided. So these images are already provided in this challenge, in this website. What you need to do is you can take these manually segmented images, and then you have to work on to identify the diabetic retinopathy in the retina. Then we have the next challenge, lesion detection. Lesion, it is a kind of a kind of a disease, or it may be some kind of basically it is some kind of disease occurring in many parts. So here you need to detect various lesions from medical images. So here they have given both CT as well as MRI images, and there are unprocessed images, that is original images, original CT and MRI images, and then they have used certain processing like contrast enhancement they have used, and they have provided the CT as well as MRI images. So they used processing as well as unprocessed image for this particular challenge. So images are available, you can take those images and you can attempt the challenges. So what are the kind of lesions they have, you are supposed to do, or you are supposed to work on? So lesion in anatomical regions such as the bones, such as liver, brain, lung, or lymph nodes. So wherever in these parts, if there is a lesion, you have to identify, you have to segment them. You have to find out where these lesions are, and you have to segment them. And to assist you, they have provided the annotated images also, that is manually annotated by the experts, medical experts. So have annotated these images, and they are being provided in this particular challenge. So you can see the website address where you can find the details of this challenge. Then we have the another problem, which is again related to the kind of endoscopy, what we call it as a colonoscopy. Colonoscopy is in this modality, the images are taken from colon. Colon means our lower part of the body. So automatic polyp detection is the kind of requirement here. So here in this, to evaluate a new and existing polyp detection algorithms. Okay, so new polyp, and just gradually if some polyp is being generated. Polyp is a kind of a tumor. You can see in this image, first image there is one tumor, second image also there is a tumor, and in third image you can clearly find there is a tumor. Okay, so you need to identify these tumors from the images that are being given. They have provided a very large set of data, okay, collected, and even there are annotated images also they are being provided. So this challenge is provided by a clinic called as Mayo Clinic in Arizona, America, and then a hospital of Universidad de Barcelona, okay, in Spain. So these two institutions jointly provided this challenge. Okay, And it's a very old challenge, 5 years old challenge. You can go through it still, but there are many new challenges also. If you want, you can attempt them. So the details are available at the website, what is being given in this slide. Then we have longitudinal multiple sclerosis lesion segmentation. So this is again related to the brain. Okay, this is a kind of multiple sclerosis lesion, is a kind of the longitudinal multiple sclerosis lesion is a kind of a disease inside the brain. Okay, you may not have heard about it. Even I was not aware of this much. So here automatic lesion segmentation algorithms has to be developed. They will provide you the MR neuroimaging data. Okay, the magnetic resonance neuroimaging data are provided here, which are acquired at multiple points of time. That means in the previous one example, we have seen first day data, 10th day data, then 28th day data, something like that. Similar kind of data are being provided over here, which are the MRI images are captured at different time periods from the same patient. So algorithm may be evaluated against manual segmentation. They have even provided the manual annotation. The experts have created done the annotation over the images, and they are being provided for a benchmark. Okay, you have to look at those images and you have to do annotation, and then you can compare your result, and you can have a, I mean, when you compare your result, your accuracy should be quite good. It should match up to 100%. So 100% matching is very ideal. At least it should match up to 95%, 98%, something like that. It should match your algorithm. So they are again the multiple raters in terms of their segmentation accuracy, and so these all they have already provided over there. The image details are available in the website that is given here. So what is the sclerosis? It is a pathological hardening or thickening of tissue. So inside that brain, that certain tissue become hard. Okay, so that is known as sclerosis. Then we have the tooth related one problem, that is BTVing. Okay, radiographic caries detection. What is this BTVing? So basically this happens due to this is a tooth related problem. Okay, so here they will give you the dental images. Okay, what is BTVing? BTVing is a dental X-ray film that can be held in places by the teeth during radiography. See, when they want to capture the X-ray, I don't know whether anybody undergone the X-ray of tooth. I have gone many a times. So they will give a small film. We have to put that film inside the mouth behind our tooth. Okay, behind our tooth, and they will put the X-ray device just in front of that film. Okay, in front of our mouth, and then they will send X-rays. So this is called as BTVing. Okay, this is BTVing. BTVing radiography. The X-ray which is captured using this particular method is known as BTVing. Then what is caries? Caries is a soft tissue decayed area in a tooth. So around the tooth, so inside the tooth there is a soft tissue. You can see that red part. Sorry, you can see this red part. Okay, this is soft tissue inside the tooth. So this is a hard tissue. That remaining part, green is very hard, yellow is next to it, and then inside that red, it is the soft tissue. Okay, so sometimes what happens, this soft tissue get decayed. Decayed means it get degraded, it reduces. Okay, so here the problem will be to identify any such degradation is happening inside the tooth soft tissue. Okay, so for that the technique what they use is they capture the X-ray image, and then they provide you the X-ray image, and then you have to analyze this X-ray image, and you have to come to decision that any degradation happens, any carie happens. Okay, caries is a progressive decay. Okay, so if the care is not taken, then the tooth will die. Die means it will get broken. Okay, and then we have to get the tooth removed by a dentist. So they provide such X-ray images collected from so many patients. They must have also provided the annotated images. So you can visit this grand challenge. You can see where actually website address is not given in the slide, but now you can see over the screen there is a website address provided. You can visit that website and you can look at the images and challenge details. Then we have the cephalometric X-ray image analysis. So what is this cephalometrics? See, you must, our skull, everyone's skull is having a different structure. Okay, the different structure means not entirely different. Small variation will be there in the everyone's skull. So basically, in the crime detection also, the police use this measurements, skull measurements, okay, for identifying whether a particular person's skull it is or not. Okay, so cephalometric is related to the head measurement, and here you need to do analysis of such head measurement. Okay, so this analysis is mandatory, providing the interpretation of patients' bony, dental and soft tissue structures and giving the whole picture of the orthodontic analysis and treatment plan making. So this is useful in the dental segment. Suppose somebody want to go for a implant, suppose somebody want to go for a fitting of the tooth sets. Okay, all these kind of things, during that time the cephalometric technique is being used by the analysts or dentists. Now the problem is they do this entirely through manual technique. Okay, so what you need to do is you have to provide your solution to automatically find generate these kind of measurements when you look at the X-ray images of the human skull. And the challenge is provided over the corner in this website. You can visit this website to identify the challenges. Then we have another problem, that is cell tracking. So cell tracking, actually this involves actually this I'm using PDF. In the if we look at PPT, actually these cells are moving. Okay, so inside body also there are certain parts where the cell movement happens. Okay, and they will provide you the videos of this sequence. You are not supposed to analyze the pictures, but you have to supposed to analyze the videos, because when you are tracking the movement, you have to work on the videos. So here tracking movement of cells in the time-lapse video sequences. So these videos are captured and we have to look at how, whether they are normally moving, whether there is an abnormal movement found in that. So your movement is measured with the time also. So which is required for many applications in both scientific and industrial setting. So this is required. So this cell may not be in the human body, but it may be in suppose for example in some due to some disease bad cells enter into our body. Okay, so there they will take the sample and then you have to analyze, or they may capture directly from that. How they capture it is unknown, but only thing is you have to analyze these cell movements. They may be captured through microscope also. They may take the sample from the body and then they can put it under the microscope, and there they can capture the movement. Okay, they can capture the video of this cell movement. So the challenge here is properly characterizing how cell moves. Okay, move as they interact with other surrounding environment. You have to look at how the cell movement is, whether normally moving, abnormally moving, fast moving, slow moving. All these kind of analysis you have to provide from this specific problem. Then we have the next, that is brain tumor image segmentation. Again, come back to the brain tumor. See, this here we are using again, I think, MRI images. See, MRI images has having so many applications. If you look at the brain itself, there are plenty of applications related to the brain itself. So in the brain image segmentation, in this challenge, whatever they have given, what you need to do is you have to segment the brain image parts into different components. You have to segment them into different parts inside the brain, and then longitudinal evaluation of time series image data. See, what is the longitudinal evaluation of time series data? The brain image, when they generate the 3D view of the brain image, 3-dimensional view, okay, how they generate? Basically, they take out the slice of the images. I will explain with a simple example. Suppose you bring a watermelon, okay, and if I look at the watermelon from outside, can I be able to identify whether watermelon is having good color, or any damage is there inside that, any disease is there inside that? Can I be able to identify? It is not possible. Okay, by looking at outside view, I can't be able to identify. Then if I want to look inside, then what I need to do? I have to cut it into small pieces. Okay, small pieces, that is slices, what we call slices. Okay, when I cut into small slices, then I can just open each slice and see, is there any disease? Is there any disease in any part? Okay, that is possible. So likewise, when we go for MRI, our actual brain, okay, entire brain is scanned slice by slice. It takes pictures slice by slice. It will not slice our brain, but it takes pictures slice by slice. Okay, and then it will generate a three-dimensional view like a watermelon. If I join them and put together, we will look again, our watermelon look like a normal. But when I open the slice, I can see what is there inside. So same thing happens in the MRI as well as CT scan. Okay, they capture every part of the slice. Again here, how much distance you want to keep. If you keep more distance, you will capture lesser part. I mean, thick parts you can capture. You will not be able to see in depth inside the image. If you keep the lesser distance of capturing, then your slice will become very thin. You will be able to analyze more in detail. Okay, but again, keeping the less, it will cost you more because you have to generate more and more MRI images. So here, then you also need to classify, okay, classify them into low and high grade gliomas. Okay, so gliomas is a kind of the tumor. You can see in the image, these tumors are visible in the image. These are CT scan images. In these CT scan images, the gliomas disease, this tumor is visible in these images. So you need to segment them, you need to classify them. There may be different types of gliomas. Okay, again, when you go to this challenge and when you look at what are the things are there, then you will be able to identify what kind of actually problems they are. So then challenge this thing, the site address is given over there. Then we will come to another brain related disease itself, what is called as dementia. Okay, Alzheimer's disease, dementia, Alzheimer's disease, dementia. So you need to, in this challenge, you need to classify these AZ dementia, Alzheimer's disease, dementia. So the problem actually is to develop algorithms that perform multi-class classification of patients with Alzheimer's disease. So they must have given the different, again, under the Alzheimer's disease, they must have given different class of diseases, or maybe they have given different levels of diseases, something like that. So you need to do classification, or they may be given the normal images and Alzheimer's disease images. So you need to classify which is good one, normal image, which is image with the Alzheimer's disease, and also you have to identify, you have to segment, you have to identify the disease part and all. Data of patients with a mild cognitive impairment, okay, and healthy controls. So they have given mild disease images as well as healthy images, both are given. Then you have to go for identification. Then multiple central structure MRI data are collected from so many one center data, because see, this is a very rare disease. Okay, Alzheimer's disease, I have not seen any patient. I have seen so many patients in the movies. Okay, but in actual life, I have not seen any patient who lose entire memory. Okay, and we see in movies and also many different types of diseases after 15 minutes losing memory and also many things we have seen. Okay, so these kind of disease are happening rarely, so you can't get enough images from any diagnostic center. So for that reason, they have collected from so many centers. So you can find the challenges. See, so much efforts are put by so many people across the world to develop these kind of data. and this is really a very useful resource for the new research people, a research fraternity. So you can find in this site the disease challenge as well as images. Then the very important part, apart from the heart and brain, inside the human body is vertebra, spine. Okay, spine is very important point. If we won't have a spine, then we won't have be able to stand, we won't have be able to walk and all, we would have gone like a snake and all. Okay, spineless. So spine is very important part of the human body. So it contains so many part, vertebra, what we lumbar, the vertebra part. So here you can find the spine, different parts of the spines. See, basically it is divided into 4 parts, you can see. Okay, the upper part, which is colored with the orange color, then we have the bluish, some different color is used in this between, that is another part. Then we have blue color, that is lower part, greenish color is there, then yellow color, that is the last part below our body. And in each part they have shown what kind of tissues are available. These are all hard tissues, these are bone tissues, okay, hard tissues. So spine segmentation algorithm you need to design. So these are all, they are providing you the CT scan images of the thoracic, okay, that is chest part, the entire spinal part they have given. Then lumbar part, what they call, that is between the rib and hip bones, okay, between rib and hip bones, and then upper part, chest and behind the chest, whatever the part we have. So these all parts images are given. You have to identify, you have to segment them, okay, because there are many diseases related to spinal cord also. So if there is sometimes some accident and all happens and spinal cord get broken, then you need to go for this kind of testing. Then we have liver ultrasound tracking. Okay, liver ultrasound tracking. So the liver ultrasound has high temporal resolution, images are given over here. Then you need to track and tracking of the motion of tissues you have to do, okay, inside the liver. So they have given you the images, you can see, and then they have colorized it. Okay, they have to colorize, they have colorized this. So you need to do tracking, and then that will help in analysis of image guided surgery and all. It will be helpful. So I will finish up. There may be few slides so that we can have some time for question and answer. Then brain imaging, what we call it as a neuroimaging. This is a very big challenge they have provided here. Many images are available over this website. You can find over there. This is a machine learning practitioner can try out their favorite or novel or not supervised learning algorithm. Supervised basically means they have already given you annotated images over here. So you have to use the annotated images as a guiding images, and then you can try out some of the algorithm development, some of the algorithms. They must have given many challenges over here. So the brain imaging, what we call it as a neuroimaging data for clinically relevant prediction problems are given here. So you can find the website where image data is given as well as the challenges are being given over here. So then we have the chest radiographic anatomical structure segmentation. So in the chest, so particularly when a person meets the accident and all, the chest bones get ribs, what we call it as, they get broken and all. So these are very helpful for that purpose. So methods for automatic segmentation of anatomical structures in chest radiographic is the challenge over here. So task start with segmentation of clavicles and later task to segment other anatomical structures. So here, see, many medical terms are being used. So when you want to take up medical image segmentation, you have to also study the medical terms and terms. Why? Because if you don't know what is this term, okay, what is there inside the chest structure, you can't be able to do it. Okay, you have to understand all these things before taking up any challenge. So here you need to identify, like a bone is given which connects both the shoulders. Okay, so that they have identified such structure identification, segmentation or problem detection is involved in this particular challenge. Then we have body labeling in 3D medical imaging. So a 3D medical image is given such as MRI and CT. Then you have to label the parts. Okay, you can see here in the images, they have labeled with the different colors, different parts of the bodies. They have labeled the chest, they have labeled the lungs, they have labeled the kidney. Okay, all tissues they have labeled, they have colored them. Okay, and then they can label it. So segmentation of anatomical structures such as the lungs, liver, kidney, or any other such tissues need to be identified, need to be segmented, and you have to label them. So already they must have provided annotated images. These are non-annotated whole body MRI and CT scan images. So you need to identify the anatomical landmarks in this data, and you have to do labeling of these parts. Image website is given over there. Then there is one mitosis detection. So mitosis, these are kind of cells which will double. Okay, so you can see in the image there is a DNA replication happening over them, and then one cell has become two cells. So to evaluate and compare, that is semi-automatic mitosis figure detection methods need to be detected, and you have to evaluate as well as compare them. This is the task involved. So works on regions extracted from whole slide images. So basically you will get the slide image in this, and you have to work on them, and then you have to perform segmentation and analysis. So since only number of mitosis figures present in the tissue is of importance, so how much tissues mitosis are there, you have to identify. What is their size and shape is not important over here. Okay, how many are there, that you need to identify in this particular challenge. So what is the mitosis? Basically it is a cell division in which the nucleus divides into nuclei containing the same number of chromosomes. So it is there, the nuclei get divided. Okay, and then same number of chromosomes are basically generated over there. So that is what the mitosis is. Then we have 3D segmentation of neuritis in electromagnetic images. So this is a full stack of electron microscope image, full stack of electron electron microscope images are given here. Then you need to do trained machine learning algorithms you have to develop for the automatic segmentation of neuritis in 3D images. So you can see here, actually this colorization is done by the computer, which has performed the segmentation. So you need to do such colorization of these neuritis from the electron microscopic images. Then we have another challenge. I think this must be the last challenge: algorithm for object classification in 3 dimensions. So a 3D image is given, maybe a brain view is given, something like that. You have to do classification. Here also they are provided images and then the data as well as images. So then segmentation of the kidney image. So algorithms for cartilage and bone segmentation from knee image. Knee images are given MRI images. You have to do cartilage and bone segmentation. Basically this technique, this challenge is launched for helping the osteoarthritis image segmentation, and that will help in planning the implant, okay, for the knee implant. Okay, we call knee implant. So for planning knee implant, these challenges are given. Then biometric measurement of fetal ultrasound image. So fetal, they have provided the growth of fetal in different weeks and all. So you have to identify biometric measurements like the length, weight, okay, something like that. Biometric identification you have to perform. Then we have coronary artery, okay, stenosis detection and quantification. So this here also a semi-automated detection and quantification of stenosis or computed tomography angiography images are provided in comparison with the quantitative coronary angiography and CTA census reading. They have provided the angiography images, and you have to perform semi-automated coronary lumen segmentation. Lumen is a darker region, okay, segmentation on these images, and you have to compare with the manually annotated image. Manually annotated images are also given. Then extraction of airway from CT. This is another challenge. I will go a little bit fast because our time remaining is very less. So here the chest, you have to develop a tree kind of structure. These images are MRI, must be CT images. CT images are given here, and you have to segment them, and you have to develop a tree airway, okay, because sometimes what happens, there may be blockages in these airways. To identify this particular technique helps us, so you can find image as well as the problem domain. Then 2D segmentation of neuronal process in EM images, so you can find more details over here. I will just move on. Then HARDI, HARDI reconstruction. This is another challenge that is being given. You can visit the website. I think website address is not given, but you can go in detail over there and you can find the challenges. So I had tried to go for so many challenges, identify so many challenges, and address some of the, provide you some of the challenges what are available. I hope my presentation is useful. If any of you have any questions, we can take up on the 10 minutes is remaining.
Sir, thank you very much, sir, for providing us such a valuable information.
Thank you.
And especially for different imaging modalities, really you have given a real research challenges, sir. Sir, there are some few questions are there in the chat box.
Oh,
so can you see
okay,
our
I will go through chat box, madam.
Okay, sir.
Okay. What is the light? Okay. What type of light will be used for biophotonics? So I think they use kind of normal kind of light, but we have to go through it in more details. We have to go through that challenge, then we will be able to identify what kind of light it is being used. So how we give the answer to pathology? Pathology, basically what I have, pathology is a external testing. Okay, suppose they take a sample and then they do pathological testing. We are not going much in that, but still we have seen certain slides there. We are taking sample and we are identifying some kind of problems, such as the one what we have looked at in the last, where the DNA replication is happening over there. So these kind of problems, even if you have pathological problem, definitely you can attempt, because such problems you have to go through grand challenges. There are hundreds of such challenges are given. I have attempted only, I have looked at only around 30 to 40 grand challenges. You can go for more challenges, maybe there you can find more and more related to pathological problems also. Can you give highlights of tools used for any software or any software used? So ready tools, if you use, then it is not a research. You have to do research. Currently, people use the MATLAB. Then if you want to use advanced tools, then you can go for a Python programming. In Python, many libraries are available to help you to do these tasks in a very shorter way. Particularly, Python is useful for deep learning algorithm development. Okay, so if you are a new researcher, then you have to do a lot of work. You have to study both the problem domain as well as you have to also study programming domain also. And then you have to also go for some deep learning course to understand what basically is deep learning, because nowadays people are using deep learning extensively. Then if you use any ready tool, then it will not serve your research problem. See how it works actually. Research, how you have to do is, you have to develop your own algorithm and then you implement. Okay, when you go for implementation, if you try to find out any readily available tool, then it will not match with your algorithm, because whatever the algorithm you have developed, you have to implement using your own programming technique. So that's why you have to go for any programming language. I recommend MATLAB if you are if you don't want to go for a difficult task. If you want to go for a very hard coding, then go for Python programming, because Python programming is very robust. It has so many libraries. Okay, it will really help you in developing good tools. How? Okay, so next participants are requested to. Okay, right. So which imaging technique is best to detect cancer in early stages? So we can't say. You have to try to develop a imaging technique which could be best one. So many techniques are being developed. Many devices have the built-in algorithms. Okay, so even you can go for a better technique. So we can't say which is best technique. It all depends on the situation and the problems. You can try out many techniques and then whichever is the best one, you can select among them. Is classification medical expert specific? Generally, it is not medical expert specific. Suppose, for example, there is a doctor related to cancer. all doctors identify same reason as a cancer. Somebody will not say that the other part is cancer. Someone else will not say that. There will be definitely there will be a difference in opinion. Okay, definitely there will be a difference in opinion. But if you look at really experts, then all of them will have same, will tell same reason as a cancerous lesion. So that's why expert, it will not be expert specific. Okay, so scholar, uh, talking up one of the challenges, taking up one of the challenges help in my research work. Surely you can see, I will put the website address also. Grand challenges, medical grand challenges. Okay, so just search there, you will find a site there. I will put the site address also in the chart. Okay, you can open this site. Many challenges are there. You can try to find from these. Actually, in fact, two of my students are also working in the medical imaging, and they have completed significant work. We have image data also. We also want to put a challenge. Okay, so these grand challenges are associated with the conferences. Okay, associated with some conferences. So they will give through conference, they will give this grand challenge, and then you have to solve them, and your paper will be get selected for that conference, and then they will even some of the grand challenges provide financial assistance also. So you can just look at many of the problems. See, for any medical imaging research, images are very, very important. Without image, you can't do any research. Okay, so image is the first thing what you need to have. So, and getting image is a very big task. Okay, that we will discuss in the next presentation. We worked on the CT images, and to get the images, we hell of the problems we faced. Okay, I will tell you what kind of problem we have faced. So image is very important. So if you want to attempt any medical imaging problems, you must have images. Without image, you can't do anything. So please remember this. Then please share the presentation. Definitely I will share. Please share all session. Okay, so can you give the tools used in AI? So see, I told you MATLAB you can use. Okay, ultimately you have to develop your algorithm. Nobody help you in developing algorithms. If you use any algorithms already readily available, then that is a it's a theft of others' work. Okay, so you should not do. You can use them, but you have to do some modification in them. You have to add to them. Value addition has to be done. If you're presenting same and telling that this is my algorithm, that is wrong. Okay, you should not do it. You have to do value addition. Okay, then only something contribution happens from your side. You can find so many open source codes also in many websites. You can find them, you can use them, no issue, but you have to use them and develop your own algorithm. That is very important. So can you please put a light on bone scan images? Okay, so I have discussed many over there. You please visit bone scanning related sites in the Grand Challenge. You can be able to find many of them. Apart from image processing challenges, can we have data analysis challenges sites suggested from your side? Okay, so this is a very good question. That is what we are going to present in the. I am going to present in my next session. We have written one paper exclusively for the data only. Okay, medical imaging data that has been published in the Journal of Medical Imaging. Sorry, it is under publication. We have communicated that to IEEE Medical Imaging. It is under review. Okay, so I will also show you that article. I will just show you. I can't share with you because it is under process of publication. So based on that, I have prepared the presentation. So this is very big task, collecting the data and analysis of the data. This is very big task. We have conducted the research on this. I will share that also with you in my next session. Very nice information, sir. Thank you for your compliments. I am working on sparse view CT image reconstruction. How deep learning techniques can be used for this? So, okay, this is also a question, good question, because see, deep learning we use during the classification. Okay, even we have used the deep learning technique in the pre-processing also. In one of my work, we have used deep learning. It is not a medical imaging work, but we have done this on the
the card classification. You know, the cards, that is the Aadhaar card, driving license or passport. So during the card pre-processing, okay, we have used deep learning technique. So suppose when you are capturing the image, you may get the background noise surrounding that this card is not in proper format, it is tilted, rotated, all these. Okay, to remove such problems, we have used a deep learning. Okay, so deep learning generally used in classification, but we have used in pre-processing techniques. Deep learning can be used. Only thing is, when you are designing the problem, you have to plan how best you can use the deep learning, particularly during the classification. This will be very, very useful. Then what is the maximum level of accuracy achieved so far for brain tumor? Please, we have not worked on any brain tumor. Please go through the grand challenges, or you can search many of the websites where people are, even the research articles, recent research articles, you can be able to identify. But in the brain tumor, there are plenty of brain tumors. Okay, which brain tumor you are talking about, that is unknown. You have to look for details. Okay, then okay, so okay, I have posted that. Then what next? I am working on sparse view CT image reconstruction. So just now I have told you, you have to, deep learning is a very good technique. So you have to learn how to, Python is best tool for deep learning. You learn Python, you go for a deep learning course. I suggest you, there are MOOC courses on the deep learning. You can go for it. In the deep learning, really future is very good, because many industries are preferring deep learning techniques. One of my student who has just recently, he has not yet submitted his thesis. A company hired him, okay, PhD student. So a company hired him because he has done wonderful work in the deep learning. Okay, now he is getting a package of 12 lakhs per annum. Okay, he has not yet submitted the thesis. He has drafted already. Shortly he will submit the thesis. So there is a great demand for deep learning techniques in the industry currently. So that is the reason for having medical experts for authentic classification. Okay.
Excuse me, sir.
Okay, madam. I think
Sir.
I have addressed most of the questions. Yes.
Sir, we'll wind up now, sir. We'll again meet back at
yes.
So request the participants, sir, will be there with us the second session. So we'll discuss there also. Okay, sir. We'll break for half an hour. Okay, thank you, sir.